All right, so we're diving into dose range finding studies today. You had some questions about this, like how do scientists actually figure out the right dose for a new drug, especially when they're using animal models?
Yeah, it's a pretty complex process.
I bet.
There's a lot that goes into it. It's not just about, finding a dose that works. It's about finding that sweet spot where it's effective, but it's also safe.
Right, you got to avoid that toxic zone, right?
Exactly. And there are all sorts of regulations and guidelines, too. It's a really fascinating area.
Okay, so let's back up for a second. Why are animal models so important in this process? I mean, before we even think about testing on humans, why do we need to go through this step with animals?
Well, you wouldn't want to test a brand new drug on a person without having any idea how it would behave in a living system.
That makes sense. Too risky. Right.
Way too risky. So animal models give us a way to study the drug in a controlled environment. It's a way to gather data on its safety and effectiveness before we even think about moving to human trials.
Makes sense. So how does that actually work? I mean, where do they even start?
Well, one of the most basic techniques is what we call an intravenous bolus.
Intravenous bolus.
Yeah, it sounds a little intimidating, but it's basically just a way to get the drug into the system quickly. You inject it right into the bloodstream.
Ah, so it bypasses all the stuff that happens when you take a pill, for example.
Exactly. And that lets us observe the drug's behavior almost immediately. We can see how it starts to work, how it distributes throughout the body.
So it's kind of like treating the body as a single compartment.
Yeah, it's a simplification, of course, but it gives us a good starting point. And to understand how a drug really behaves, we need to look at something called pharmacokinetic parameters.
Pharmacokinetic parameters.
Yeah, think of these as our detectors for drug behavior.
Okay.
They tell us how the drug is absorbed, distributed, metabolized, and then eliminated from the body.
Our little drug detectives.
Oh.
Okay, so tell me more about these detectives. What kind of clues are they looking for?
All right, so one of the key players here is the apparent volume of distribution. It's often represented by the letter V.
V for volume, right?
Yeah, but it's not a volume you can actually measure, like with a ruler or something. It's more of a mathematical tool.
Okay, so it's not like a literal
But
volume.
no, it's more about understanding where the drug goes in the body, like how much of it stays in the bloodstream and how much of it goes into the tissues.
Got it.
Think of it this way. Imagine you have a beaker of water with charcoal in it.
Okay.
If you add some kind of substance to that water, some of it will bind to that charcoal, right?
Yeah.
And that changes the overall concentration of the substance in the water.
Makes sense. So it's kind of like that.
Yeah, the V value helps us understand how much of a drug binds to tissues, which tells us how it's distributed throughout the body.
Okay, that's a good analogy. So a high V would mean the drug is spreading out a lot.
Exactly. And a low V would mean it's mostly staying in the blood.
All right, got it. So what other detective clues are we looking for?
Well, there's also the elimination half-life, or T. This one's all about time.
Time.
Yeah, it tells us how long it takes for half of the drug to be eliminated from the body.
Oh, okay. So
Right.
that would affect how often you need to take it, right? Like if it gets eliminated quickly, you need to take it more often.
Exactly. And that's why T is so important in determining the right dosing schedule. And then we have the elimination rate constant, or K.
Okay.
Yeah, think of this one as the speed at which the drug exits the body.
Okay, so high K, it's leaving quickly.
Exactly. And those usually go hand in hand with a shorter half-life.
Right. It all makes sense. They're all connected. So we've got our detectives, we've got our clues, but how do they actually figure out the right dose range? How do they put all of this together?
Well, that's where things get really interesting. They track the drug's concentration in the blood over time, and they use those pharmacokinetic parameters, like V, T, and K, to build a mathematical model.
A model.
Yeah, it's like a simulation of how the drug is behaving in the body. And that model helps them predict what will happen at different doses. They can use it to calculate an initial safe dose range.
So they're not just guessing, they're actually building a model to figure this out.
Exactly. And of course, there are tons of regulations and guidelines to make sure all of this is done ethically and consistently. The FDA and the ICH, they're all over this.
That makes sense, right? Huge responsibility. So it's not just about the science. It's about protecting the animals and ultimately the humans, too.
Absolutely. It's about laying a solid foundation for drug development where safety and effectiveness are the top priorities.
This is fascinating. But I'm curious, how do they figure out the actual dosing schedule? Do they give it all at once, spread it out? How does that work?
That's a great question, and it's where things get even more interesting. The dosing schedule can make a big difference in how well a drug works and how safe it is. Maybe we can dive into those different approaches after a quick break.
Yeah, let's do that. We'll be right back to explore all those different dosing strategies.
So we talked about how those pharmacokinetic detectives like V, T, and K help us figure out that starting dose range. But the dosing schedule, that's another big piece of the puzzle.
Yeah, I was thinking about that, like giving a drug once a day versus, I don't know, multiple times a day. That seems like it would make a huge difference.
Oh, absolutely. The timing and frequency of those doses can really impact how the drug behaves in the body. Think about it like cooking, right? Sometimes you throw everything in the pot at once, and sometimes you have to add ingredients slowly.
I like that analogy. So walk me through some of the different dosing schedules they might try in these studies.
Well, one approach is what's called a single dose study. They give the animal just one dose and then carefully monitor what happens. It's like taking a snapshot of the drug's behavior at that specific time.
Okay, that makes sense for getting an initial understanding. But what about long-term effects? How do they study that?
For that, they might use a repeated dose study. So, giving the drug multiple times over days, weeks, maybe even months.
So like a time-lapse video instead of a single picture.
Exactly. And that helps them understand how the drug builds up in the body over time. They can see if there are any, cumulative effects or any signs of toxicity.
So it's not just about the dose itself, it's about how often you give it too.
Right. And within those repeated dose studies, there are different variations, like giving the drug once a day, twice a day, or even more often. It all depends on the drug's half-life and what kind of effect they're looking for.
Makes sense. A short half-life, you need to give it more frequently to keep it working.
Exactly. It's like tailoring a suit. You need the right measurements for it to fit properly. And it's not just about how often, but also the route of administration. We've talked about intravenous bolus, but you can give drugs orally, intramuscularly, subcutaneously, even through inhalation.
Wow, so many options. It seems like there are a lot of factors to consider. But how do they actually measure the outcomes? What are they looking for to know if the drug is safe and effective?
Well, they use a whole range of outcome measures for both efficacy and toxicity. On the efficacy side, they might look for changes in biomarkers or improvement in behavioral tests. If it's a cancer drug, they might measure tumor size.
So it's tailored to what they're trying to treat.
Exactly. And for toxicity, they're looking for any signs of adverse effects, like changes in blood chemistry or even organ damage.
It's a delicate balancing act, isn't it? Trying to get the good effects without causing any harm.
That's exactly it. And that's why these preclinical studies are so important. They're the foundation for those clinical trials in humans.
Now, I remember you mentioned earlier that intravenous bolus is kind of like a starting point because it bypasses the complexities of absorption. But how do they factor in different routes of administration when they're figuring out the dose and schedule? I mean, a pill is going to be different from an injection, right?
That's a great point. It's true that IV is the most direct route, but often a drug will be given orally or some other way. So they do have to account for those differences.
So how do they do that? How do they adjust for the fact that, say, a pill might not be absorbed as well as an injection?
Well, there's this concept called bioavailability. It basically refers to how much of the drug actually gets into the bloodstream. With 5E, it's pretty much 100% because you're putting it directly into the bloodstream.
But with a pill, some of it might get broken down before it even gets there.
Exactly. And that can vary depending on things like the drug's formulation, whether it's taken with food, and even individual differences in metabolism.
So if a drug isn't absorbed well when taken orally, they might need to give a higher dose to get the same effect.
That's right. And this is where those pharmacokinetic parameters come back into play. They help us predict how the drug will behave based on the route of administration.
Wow, this is really complex. I'm starting to see how all these pieces fit together. Like, you have to think about the route, the bioavailability, the half-life, the volume of distribution. It's a lot.
It is. It's like fine-tuning an instrument. And researchers use all sorts of tools, mathematical models, lab techniques, to figure out how to dose a drug effectively.
This has been so insightful. We've talked about animal models, dosing schedules, outcome measures. It's amazing how much goes into this research. But I'm curious, are there any limitations, any challenges they face in these studies?
That's a great question, and it speaks to how this field is always evolving. Maybe we can explore some of those challenges after a quick break. We'll be right back.
Sounds good. We'll be back in a moment to continue our deep dive into the world of dose range finding studies.
So you asked about challenges in these dose range finding studies. Well, one of the biggest is, animal models and humans, they're not exactly the same.
Right, a mouse isn't a tiny human.
Yeah, exactly. What works in a mouse might not work the same way in a human.
Of course.
There can be differences in how the drug is absorbed, how it's broken down, even how it's eliminated from the body. And then, even within a species, you have variations like age, sex, genetics, all those things can play a role.
So you can't just assume that what you see in an animal study will perfectly translate to humans.
Exactly. It's like, well, it's not a perfect one-to-one comparison. And that's why those regulations we talked about, those are so crucial. Having standardized methods, rigorous controls, all of that helps minimize those variations and makes the data more reliable.
So it's about controlling what you can control.
Yeah.
Are there other limitations they have to think about?
Oh, absolutely. There's the ethical aspect of using animals in research. Animal research is essential for medical progress, but it has to be done ethically. We have a responsibility to those animals.
Yeah, it's a tough balance, isn't it? The need for this research versus the well-being of the animals.
Exactly. And that's why there's this big push for what we call the 3 Rs: replacement, reduction, and refinement.
The 3 Rs. Tell me more about those.
So replacement means finding alternatives to using animals whenever possible, like cell cultures or computer models. And reduction means using the fewest animals possible while still getting good data. And then refinement, that's all about improving the experiments themselves to reduce any pain or distress for the animals.
So it's about constantly finding ways to do things better, more humanely.
Exactly. And those ethical considerations, they're part of every step, from choosing the right animal model to how the experiments are designed to making sure the animals are well cared for.
That's good to hear. You mentioned that this field is always evolving. So are there any new technologies or approaches that might change how they do these studies in the future?
Oh, yeah, definitely. One really exciting area is these in vitro models that are becoming more and more human relevant. They use human cells or tissues grown in the lab, so you're getting closer to studying the drug's effects in a human system.
Bringing the human element into the lab.
Exactly. And then there's computer modeling. That's becoming a big deal. Researchers can create virtual models of the body and test how different drugs might behave, potentially reducing the need for some animal studies.
Wow, so technology is opening up a whole new world for this kind of research.
It really is. And as those technologies keep advancing, we're going to see even more precise and efficient ways to figure out the right dose for new drugs.
This has been incredibly insightful. We've covered so much ground, from those little drug detective parameters to the complexities of dosing schedules and outcome measures, and of course the ethical side of things, which is so important. What do you think is the most important thing for people to take away from this conversation?
I'd say it's that balance, that constant balancing act between wanting the drug to work but also wanting it to be safe. It's like walking a tightrope, trying to find that sweet spot where the benefits outweigh the risks. And honestly, it's really amazing what these researchers are doing, pushing the boundaries of science to find new treatments while also being mindful of the ethical implications.
It really is remarkable work. And I think we've all learned a lot about the science behind it, the challenges, and the ethical considerations that are so important. As you continue to think about all of this, maybe consider this. How do you think personalized medicine, tailoring treatments to individuals, how might that impact the future of dose range finding studies? That's something to think about.
Oh, that's a great point. Yeah, that idea of personalized medicine, that could really shake things up. It shows just how dynamic this field is. The quest to find safe and effective treatments, it's an ongoing journey, but with all these advancements, I think the future of drug development is incredibly bright.
Well, thanks for joining us on this deep dive into dose range finding studies. Until next time, keep asking those questions and keep exploring.